In this video, we will talk in detail about the structures of DNA and RNA. DNA and RNA are both composed of three main components. The first component is a 5-carbon sugar, pictured here in the center. Now DNA, its sugar is called deoxyribose, whereas RNA has a different sugar called ribose. So that's where those names come from. DNA for deoxyribose, RNA for ribose. The second main component of nucleotides, DNA and RNA, is a phosphate group. And the third component, pictured up here, are the nucleotide bases. So there are actually 5 different nucleotide bases. For DNA, the bases are A, T, C, and G, whereas RNA, the bases are A, U, C, and G. So as you can see here, A is for adenine, G for guanine. Adenine and guanine both have these 2 ring structures. So there's 2 rings that are fused together. If the molecule, the nucleotide, has 2 rings, we call this a purine. Whereas cytosine, C, thymine, which is in DNA, and uracil, which is in RNA only, they have this 1 ring structure. These are called pyrimidines. A purine always pairs with a pyrimidine. So, for example, in DNA, A always pairs with T, and C always pairs with G. So A, which is a purine, pairs with T, which is a pyrimidine, and C, which is a pyrimidine, pairs with G, which is a purine. Whereas for RNA, A isn't going to pair with T anymore, it's going to pair with U. Here is what the phosphate, if we go back to the previous slide, remember phosphate is the second major component of DNA and RNA. This is what the phosphate looks like. Then lastly, the third component we said was that 5-carbon sugar, deoxyribose, which is in DNA, and ribose, which is in RNA. And if you really look at these 2 structures, you can see they're basically identical to one another. The only difference is this part right here. Ribose has an extra oxygen here. Deoxy means lost an oxygen. So DNA is missing that second oxygen right here. So deoxyribose versus ribose. Now, let's talk about how these components, okay, phosphate, sugar, and the base, how do they come together? All right. So what is really important is when you look at ribose and when you look at deoxyribose, they both have 5 carbons. And we number them in a specific order, such as this, okay? The oxygen is always here at the top. Carbon 1 is always this carbon that's right next to the oxygen. So this one here is carbon number 1. The next one is 2, carbon 3, 4, and now carbon 5 is the one that's kind of sticking out at the end here. So the way we write these numbers is we don't just say 1, 2, 3, 4, 5. We say 1 prime. Okay, so this little symbol here, this means prime. So this is the 1 prime carbon, the 2 prime carbon, 3 prime, 4 prime, and 5 prime. So when you see these little symbols next to the numbers, that means prime. 1 prime, 2 prime, 3 prime, 4 prime. Now, the base, okay, the A, T, C, G, or U is always attached to the 1 prime carbon. Okay, so the base will attach at 1 prime. The phosphate group will always attach to the 5 prime carbon. Okay, so phosphate is attached at the 5 prime. Now, in real DNA, we don't just have one of these, we have many of these that are going to link together. Okay, so for example, here's one group with a base, the sugar and the phosphate, but they're going to link to a second one, and it's going to link to a third, and we're going to make a chain, right? A polynucleotide chain with a bunch of these that are all going to stick together. So here's what this is going to look like, and I know my drawings are really bad, especially on this, but bear with me, okay? So you have your 5 carbon sugar here, okay? 1 prime carbon, 2 prime carbon, 3, 4, and 5. All right, and then you have your base that's going to stick to the 1 prime carbon. And then you have the phosphate group, which is going to stick to the 5. All right, and now you have another one of these that's coming in. All right, so I'm going to draw a second one of these. Now what's going to happen is this phosphate is going to link up with this sugar here. All right, so this forms a new bond. Now this bond has a special name. We call it a phosphodiester bond. So phosphodiester bonds link phosphates with the 5 carbon sugar. All right, they're going to link the phosphate with the 5 carbon sugar. Now, as you know, DNA is an extraordinarily strong molecule. So these phosphodiester bonds are very strong. They are a type of covalent bond. Remember, covalent bonds, electrons are shared and they're extraordinarily strong bonds. So in real DNA, what you're going to have is a long chain of these molecules that all stick together. And on the outside of the DNA, you're going to have alternating phosphates, sugars, phosphate, sugar, phosphate, sugar. All right, so we call this the phosphate, sugar-phosphate backbone, which is the backbone of DNA. Okay, now there's one more thing to note here. Eventually, there's no more DNA to add and we've hit the end here, okay? So on one end, you can see there's a phosphate here at this end, but at the other end, there's no phosphate. We're done, okay? We've reached the end. The end that has this phosphate molecule, this is called the 5 prime end. The reason we call it the 5 prime end is because phosphate attaches to 5, carbon 5, so this 5 is at the end here, and this phosphate is exposed. So you always know it's the 5 prime end when you see an exposed phosphate at the very end of the DNA. The opposite end, there's no phosphate here. Remember, this is carbon 1, 2, this is 3, okay? So this end we call the 3 prime end. So the 3 prime end is the one where there's no phosphate. And you just see the sugar is at the end of the molecule. All right. So if we go back to our PowerPoint here, you can see this alternating phosphate, sugar. This end is the 5' end of the strand, whereas this end, where you see there's no phosphate, but there's an exposed OH here, this is the 3' end of the strand. The bases, the nucleotide bases, will stay in the middle of the DNA molecule. Now you know that DNA is not single-stranded. DNA is a double-stranded molecule, which means that the bases are going to pair up in the middle of the DNA helix. All right? And the bases are what we call complementary. And what this basically means is that the A always pairs with the T, the C always pairs with the G. It goes the reverse too. T always pairs with A, C always pairs with G. These bases are held together by a different type of bond called a hydrogen bond. These bonds are relatively weak. Now here's something that's really important. DNA is not only are the strands going to be complementary, the strands are also what we call anti-parallel. Okay? So what this means is on one strand, here you can see the phosphates at the end, so you know this is the 5' end. The other side where you see this exposed hydroxyl group is the 3' end. Now when two strands come together here, okay, you see this complementary base pairing, the second strand is going to be the opposite, in the opposite direction of the first. And this is what anti-parallel means. It means the two strands are opposite of each other. So on this strand, where you see the 5' end, the opposite strand is 3'. All right, you see an exposed hydroxyl group here. And on this end where we had the 3' end, this opposite strand is 5'. So you can see 5 and 3 are always opposite of each other. And this makes DNA anti-parallel. So let's review. What are the 4 nucleotide bases? These are the ones found in DNA. They are A, which pairs with T, adenine pairs with thymine, and cytosine, which pairs with guanine. What does it mean that DNA is anti-parallel? It means that the two strands are opposite. They're in reverse of each other. All right, and basically what that always means is 5' end is always opposite the 3' end. What does it mean that two DNA strands are complementary? This means that the bases that are lined up in the center of the DNA strand will pair appropriately, either A to T or C to G. What are the 3 components of DNA? We know they are the base, the nucleotide base, the deoxyribose sugar, and the phosphate group. Lastly, let's talk quickly about RNA. Okay, RNA is just like DNA, only a few differences. Remember, we have U instead of T in RNA. And RNA is always single-stranded. It doesn't need that double strand like DNA. And lastly, RNA uses ribose sugar instead of deoxyribose. Just like DNA has a 5' and a 3' end, RNA also has a 5' and a 3' end. So if you looked at this picture, you would know here's an exposed phosphate. This has to be the 5' end. Down here, there's no phosphate. All right, the sugar's at the end. So you know this is the 3' end.